Hello, my name is Karlo Díaz. I am studying medicine in the Bolivarian Pontifical University. Today I go to explain you about the characterization of single amino acid variation in a EDTA tolerating non-specific nucleases from the ICE nucleating bacterium Pseudomonas syringae. Introduction. What is a pseudomonad? A pseudomonad is a genus of straight or slightly curved bacilli, gram-negative, strict aerobic, and oxidase positive, but also, or in some cases, they can use nitrates as an electron acceptor. The Pseudomonas syringae is very important in this article because it's very resistant, have a capacity to infect the plants, causing infections, obviously, and that determines significant economical losses, production, and the equality. Overall objective is characterizes the single amino acid variations in a non-specific nucleases from the ICE nucleating bacterium Pseudomonas syringae. Methods. Cloning. Cloning is a method used for create identical copies of the DNA original. First, you have to acquire the plasmid that contains the DNA that you have to cloning and insert this plasmid into a bacterium. The bacterium have the mechanism for replicate this DNA, but you need restriction and the nucleases specific for this DNA. In this case, the cloning was used for generate plasmid, this plasmid basically, recombinant expression. When you put the DNA into a bacterium, the bacterium starts the process of replication, transcription, and traduction, forming proteins. That proteins are very important because this article require the proteins or the enzyme, in specific the nucleases, for the quantify. Okay, if you want to quantify the proteins in the solution or in the bacteriums, do you have to lysing these bacteriums and obtain the proteins. Later, you have to use the electrophoresis. The electrophoresis is a method used for quantify the charge and the weight in protein, DNA, or RNA. Constants of a platform with a gel. The gel is a polysaccharide, generally saccharosa. Kinetic. The kinetic is important because this determines the function and the activity of the protein in the organism, in the bacterium, for example. Results. The figure 1, we can see the difference in a control and activity of the enzyme or the nucleases, and we can see that the molecular weight in the nuclease is approximately 70 kilodaltons. Next, substrate specificity EDTA and salt threat and influence of temperature and pH on the activity of PSNAC. We can see the different activity of the same nuclease have a lot of power without all the types or nucleotides. And here we can see the resistance of the nuclease at the EDTA. Here we can see the evolutionary basis genetic variant of the nucleases. Here we can see variants of the nuclease, and these variants are inefficient. Here we can see the proteins that aren't degraded or has not a correct function. And the conclusion. For my conclusion, it's the non-specific nucleases of pseudomonas are very tolerant enzymes to adverse environment. All non-specific nucleotides, not all non-specific nucleotides variants of the pseudomonas have the same tolerances capabilities against EDTA.